What are green-beard genes? To explain this term, we must start from the very beginning. Charles Darwin hypothesized the theory of natural selection where individuals that have advantageous traits are more likely to survive and reproduce, producing offspring that have the same traits, given that the traits are heritable. This was followed by Ronald Aylmer Fisher, who proposed natural selection from the perspective of genes, where possession of genes that increased fitness of an individual results to an increased frequency of those same genes in the gene pool. However, in the 1960s, William Donald Hamilton realized that altruistic behavior could not be explained by the hypotheses proposed by Darwin and Fisher. An altruistic behavior is one that benefits the receiver of the action but is often detrimental to the actor. If so, why has this trait not been removed by natural selection? The answer was proposed by Hamilton. He demonstrated that genes that increased fitness of an individual can spread both by producing offspring with those genes or by ensuring that other individuals do through altruistic behaviors directed towards them. Now this leads to the question of how they will know whether another individual is carrying that same gene. Hamilton then proposed that there could be a gene which gives rise to a phenotype that others with that same gene are able to recognize and direct altruistic behaviors towards. This gene was then given the name green-beard gene by Richard Dawkins. However, it is important to note that the phenotype may not be a visible one. Although this hypothesis does provide a mechanical explanation for the existence of altruistic behavior, many thought that the chances of finding a real green-beard gene was close to none due to many reasons. One of them is that such genes are likely to be fixed in a population during natural selection. This means that every individual will possess a green beard, and so individuals will not manifest a conditional social behavior. This was until Laurent Keller and Kenneth G. Ross discovered the GP9 gene in fire ants Solenopsis invicta. Like any gene, there are two alternative alleles at the GP9 locus, and they are denoted as big B and little b, resulting to three possible genotypes: big B, big B, big B, little b, or little b, little b. The experiment Keller and Ross conducted focused on polygynous fire ant colonies where there are multiple queens. In 1997, Ross found that females that have the little b genotype died prematurely, preventing the fixation of the little b allele in the population. In 1998, together with Keller, they found that heterozygous queens were the primary reproducers of the colony. So what about the big B queens? Well, they were murdered. Keller and Ross reintroduced young reproductive queens into the original polygynous nest, and the results obtained were shocking. They found that 61% of big B queens that were 7 to 10 days old and 91% that were 11 to 14 days old were attacked by workers. In both age groups, none of the queens carrying a little b allele were attacked. This suggested that the probability of queens being attacked by workers was related to the genotype at the GP9 locus. So Keller and Ross did a little investigation to find the culprits. They counted the number of worker ants surrounding the attacked queens and identified their genotype. Lo and behold, approximately 78% of them were workers carrying the little b allele. But how did they know who to attack? Keller and Ross noticed that a couple of workers that attacked the big B queens were also attacked by other workers. So they took cuticles of big B and big B little b queens and stroked them against random worker ants. Interestingly, those that were rubbed against cuticles of big B queens expressed more aggression than those rubbed against cuticles of big B little b queens. Keller and Ross then concluded that workers carrying the little b allele can recognize chemical cues that 1. Indicates sexual maturity of a queen as proposed by David Fletcher and Murray Blum, and 2. Her genotype at the GP9 locus. With Keller and Ross's discovery, Dawkins's green-beard thought experiment was proven to exist in nature. However, the GP9 gene was only one of 4 types named facultative-helping green-beard gene. Following Keller and Ross, scientists were later able to discover facultative harming, obligate harming, and obligate helping green-beard genes. Of course, studies on green-beard genes have only just begun, and there are still many questions left unanswered about them.